What is TCA cycle? The tricarboxylic acid cycle is also known as the Krebs cycle or the citric acid cycle. It is the central metabolic pathway responsible for oxidizing acetyl A derived from carbohydrates, proteins, and fats. The tricarboxylic acid cycle occurs in the mitochondrial matrix of eukaryotic cells. Now, what is the purpose of the tricarboxylic acid cycle? 1. Energy production. It oxidizes acetyl A to produce high-energy electron carriers, which are NADH and FADH. 2. Cellular respiration. These high-energy electron carriers produced are passed into the electron transport chain to generate ATP. 3. Biosynthesis. It provides precursors for building of amino acids, fatty acids, and nucleotides. Now, steps of the TCA cycle. In step 1, the 2-carbon acetyl A combines with a 4-carbon oxaloacetate, releasing the CoA group and forming a 6-carbon molecule called citrate. In step 2, citrate is converted to its isomer, isocitrate. This happens in a 2-part process where a water molecule is first removed and then added back, and this 2-step reaction is catalyzed by the enzyme aconitase. In step 3, isocitrate is oxidized, releasing a molecule of carbon dioxide. Simultaneously, NAD+ is reduced to NADH. This is a rate-limiting step of the cycle and catalyzed by the enzyme isocitrate dehydrogenase. In step 4, alpha-ketoglutarate is oxidized, releasing another molecule of carbon dioxide. Another NAD+ is reduced to NADH, and it binds to coenzyme A to form a 4-carbon succinyl CoA. This step is catalyzed by the enzyme alpha-ketoglutarate dehydrogenase. In step 5, the CoA is replaced by a phosphate group and energy is transferred to form GTP. This leaves a 4-carbon molecule called succinate, and this step is catalyzed by the enzyme succinyl CoA synthetase. In step 6, succinate is oxidized to fumarate. 2 hydrogens are transferred to FAD, creating FADH2, and this step is catalyzed by the enzyme succinyl CoA dehydrogenase. In step 7, a water molecule is added to fumarate to convert it into malate, and this step is catalyzed by the enzyme fumarase. And in step 8, malate is oxidized back into oxaloacetate, regenerating the cycle's third NADH molecule and allowing the cycle to repeat. Now, this step is catalyzed by the enzyme called malate dehydrogenase. Now, we are going to see some key electron carriers in the TCA cycle. NAD stands for nicotinamide adenine dinucleotide. Now, this is the oxidized form of this coenzyme. It acts as an electron acceptor during glycolysis and TCA cycle. And NADH is the reduced form of NAD, which has accepted high energy electrons and hydrogen atom. Now, FAD stands for flavin adenine dinucleotide, which is also an electron acceptor similar to NAD in the TCA cycle. Now, FADH and FADH2 is the reduced form of FAD, which has accepted 2 electrons and 2 protons. Now, if you found this lesson helpful, please like this video and subscribe for more simplified biochemistry lessons. Thank you.